Chapter 12 Mineral Nutrition. The basic needs of all living organisms are essentially the same. They require macromolecules such as carbohydrate, protein, and fats, and water and minerals for their growth and development. This chapter focuses mainly on inorganic plant nutrition, wherein you will study the methods to identify elements essential to growth and development of plants and the criteria for establishing the essentiality. You will also study the role of the essential element, their major deficiency symptoms, and the mechanism of absorption of these essential elements. The chapter also introduces you briefly to the significance and the mechanism of biological nitrogen fixation. 12.1 Method to study the mineral requirement of plants. In 1860, Julius von Sack, a prominent German botanist, demonstrated for the first time that plant could be grown to maturity in a defined nutrient solution in complete absence of soil. This technique of growing plant in a nutrient solution is known as hydroponics. Since then, a number of improvised methods have been employed to try and determine the mineral nutrients essential for plants. The essence of all these methods involve the culture of plant in a soil-free, defined mineral solution. These methods require purified water and mineral nutrient salts. Can you explain why is this so essential? After a series of experiments in which the roots of the plant were immersed in nutrient solution and wherein an element was added, substituted, removed, or given in varied concentration, a mineral solution suitable for the plant growth was obtained. By this method, essential elements were identified and their deficiency symptoms discovered. Hydroponics has been successfully employed as a technique for the commercial production of vegetables such as tomatoes, seedless cucumber, and lettuce. It must be emphasized that the nutrient solution must be adequately aerated to obtain the optimum growth. What would happen if solution were poorly aerated? Diagrammatic views of the hydroponic technique is given in figure 12.1 and 12.2. 12.2 Essential mineral element. Most of the mineral present in soil can enter plants through root. In fact, more than 60 elements of 105 discovered so far are found in different plants. Some plant species accumulate selenium, some other gold, while some plants growing near nuclear test site take up radioactive strontium. There are techniques that are able to detect the minerals even at very low concentration, that is 10 to the power minus 8 gram per milliliter. The question is whether all the diverse mineral elements present in a plant, for example gold and selenium as mentioned above, are really necessary for plants. How do we decide what is essential for plants and what is not? 12.21 Criteria for essentiality. The criteria for essentiality of an element are given below. A. The element must be absolutely necessary for supporting normal growth and reproduction. In the absence of the element, the plants do not complete their life cycle or set the seeds. B. The requirement of the element must be specific and not replaceable by another element. In other words, deficiency of any one element cannot be met by supplying some other element. C. The element must be directly involved in the metabolism of the plant. Based upon the above criteria, only a few elements have been found to be absolutely essential for plant growth and metabolism. These elements are further divided into two broad categories based on their quantitative requirement. First, macronutrient, and second, micronutrient. Macronutrients are generally present in plant tissue in large amount in excess of 10 millimole per kg of dry matter. The macronutrients include carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur, potassium, calcium, and magnesium. Of these, carbon, hydrogen, and oxygen are mainly obtained from CO2 and H2O, while the other are absorbed from the soil as mineral nutrition. Micronutrition nutrients are trace elements are needed in very small amount, less than 10 millimole per kg of dry matter. These include iron, magnesium, copper, molybdenum, zinc, boron, chlorine, and nickel. In addition to the 70 essential elements named above, there are some beneficial elements such as sodium, silicon, cobalt, and selenium. They are required by higher plants. Essential elements can also be grouped into four broad categories on the basis of their diverse function. These categories are: first, essential elements as components of biomolecule and hence structure elements of cell. Example: carbon, hydrogen, oxygen, and nitrogen. Second, essential elements that are components of energy related chemical compound in plants. Example: magnesium in chlorophyll and phosphorus in ATP. Third, essential element that activate or inhibit enzyme. For example, magnesium plus 2 is an activator for both ribulose biphosphate carboxylase oxidase and phosphoenol pyruvate carboxylase, both of which are critical enzyme in photosynthetic carbon fixation. Zinc plus 2 is an activator of alcohol dehydrogenase and molybdenum of nitrogenase during nitrogen metabolism. Can you name a few more elements that fall in this category? For this, you will need to recollect some of the biochemical pathway you have studied earlier. Some essential elements can alter the osmotic potential of a cell. Potassium play Point 4. Some essential elements can alter the osmotic potential of a cell. Potassium play an important role in the opening and closing of stomata. You may recall the role of mineral as solutes in determining the water potential of a cell. 12.22 Role of macro and micronutrients. Essential elements perform several functions. They participate in various metabolic processes in the plant cell, such as permeability of cell membrane, maintenance of osmotic concentration of cell sap, electron transport system, buffering action, enzymatic activity, and act as a major constituent of macromolecule and coenzyme. Various forms and function of essential nutrient element are given below. Nitrogen. This is the essential nutrient element required by plants in the greatest amount. It is absorbed mainly as NO3 minus, though some are also taken up as NO2 minus or NH4 plus. Nitrogen is required by all parts of a plant, particularly the meristematic tissue and the metabolically active cell. Nitrogen is one of the major constituent of protein, nucleic acid, vitamins, and hormones. Phosphorus is absorbed by the plant from soil in the form of phosphate ions, either as H2PO4 minus or HPO4 minus 2. Phosphorus is a constituent of cell membrane, certain protein, all nucleic acid and nucleotides, and is required for all phosphorylation reaction. Potassium. It is absorbed as potassium ion in plant. This is required in more abundant quantities in the meristematic tissue, buds, leaf, and root tips. Potassium helps to maintain an ion cation balance in cell and is involved in protein synthesis, opening and closing of stomata, activation of enzyme, and in the maintenance of the turgidity of cells. Calcium. Plant absorb Plant absorb calcium from the soil in the form of calcium ions, that is Ca plus 2. Calcium is required by meristematic and differentiating tissue. During cell division, it is used in the synthesis of cell wall, particularly as calcium pactate in the middle lamella. It is also needed during the formation of mitotic spindle. It accumulates in older leaves. It is involved in the normal functioning of the cell membrane. It activates certain enzymes and play an important role in regulating metabolic activities. Magnesium. It is absorbed by the plants in the form of divalent magnesium plus 2. It activates the enzyme of respiration photosynthesis and are involved in the synthesis of DNA and RNA. Magnesium is a constituent of the ring structure of chlorophyll and helps to maintain the ribosome structure. Sulfur. Plants obtain sulfur in the form of sulfate, that is SO4 minus 2. Sulfur is present in two amino acids, cytosine and methionine, and is the main constituent of several coenzyme vitamins, that is thiamine, biotin, coenzyme A, and ferredoxin. Iron. Plant obtain iron in the form of ferric ions, that is Fe plus 3. It is required in the larger amount in comparison to the other micronutrient. It is an important constituent of protein involved in the transfer of electrons like ferredoxin and cytochromes. It is reversibly oxidized from Fe plus 2 to Fe plus 3 during electron transfer. It activates catalase enzyme and is essential for the formation of chlorophyll. Manganese. It is absorbed in the form of manganese ion, that is Mn plus 2. It activates many enzymes involved in photosynthesis, respiration, and nitrogen metabolism. The base defined function of manganese is in the splitting of water to liberate oxygen during photosynthesis. Zinc. Plant obtains zinc as Zn plus 2 ions. It activates various enzymes, especially carboxylases. It also needed in the synthesis of oxygen. Copper. It is absorbed as cupric ion, Cu plus 2. It is essential for the overall metabolism in plant. Like iron, it is associated with the certain enzymes involved in redox reaction and is reversibly oxidized from Cu plus to Cu plus 2. Boron. It is absorbed as BO3 or B4O7 minus 2. Boron is required for uptake and utilization of Ca plus 2. Membrane functioning, pollen germination, cell elongation, cell differentiation, carbohydrate translocation. Molybdenum. Plants obtain it in the form of molybdate ions, MoO2 plus 2. It is a component of several enzymes, including nitrogenase and nitrate reductase, both of which participate in nitrogen metabolism. Chlorine. It is absorbed in the form of chlorine anion, that is Cl minus, along with Na plus and K plus. It helps in determining the solute concentration and the anion cation balance in cell. It is essential for the water splitting reaction in photosynthesis, a reaction that leads to oxygen evolution. 12.23 Deficiency symptoms of essential element. Whenever the supply of an essential element becomes limited, plant growth is retarded. The concentration of the essential elements below which plant growth is retarded is termed as critical concentration. The element is said to be deficient when present below the critical concentration. Since each element has one or more specific structural or functional role in plants, in the absence of any particular element, plants show certain morphological changes. These morphological changes are indicative of certain element deficiencies and are called deficiency symptoms. The deficiency symptoms vary from element to element and they disappear. Deficiency symptoms vary from element to element and they disappear when the deficient mineral nutrient is provided to the plant. However, if If deprivation continues, it may eventually lead to death of the plant. The parts of the plant that show the deficiency symptoms also depend on the mobility of the element in the plant. For elements that are actively mobilized within the plants and exported to young developing tissue, the deficiency symptoms tend to appear first in the older tissue. For example, the deficiency symptoms of nitrogen, potassium and magnesium are visible first in the senescent leaf. In the older leaf, and biomolecules containing these elements are broken down, making these elements available for mobilizing to younger leaves. The deficiency symptoms tend to appear first in the young tissue whenever the elements are relatively more immobile and are not transported out of the mature organs. For example, elements like sulfur and calcium are a part of the structural component of the cell and hence not easily released. These aspects of mineral nutrition of plant is of a great significance and importance to agriculture and horticulture. The kind of deficiency symptoms shown in plants include chlorosis, necrosis, stunted plant growth, premature fall of leaves and buds, and inhibition of cell division. Chlorosis is the loss of chlorophyll leading to the yellowing in leaves. This symptom is caused by the deficiency of element N, K, Mg, S, Fe, Mn, Zn and Mo. Likewise, necrosis or death of tissue, particularly leaf tissue, is due to the deficiency of Ca, Mg, Cu, lack or low level of NKS, MO cause an inhibition of cell division. Some elements like NSMO delay flowering if their concentration in plants is low. You can see from the above that the deficiency of any element can cause multiple symptoms and that the same symptoms may be caused by the deficiency of one of several different elements. Hence to identify the deficient element, one has to study all the symptoms developed in all the various parts of the plant and compare them with the available standard tables. Sorry. We must also be aware that different plants also respond differently to the deficiency of the same element. 12.24 Toxicity of micronutrient. The requirement of micronutrient is always low in low amount, while their moderate decrease causes the deficiency symptoms and a moderate increase causes toxicity. In other words, there is a narrow range of concentration at which the elements are optimum. Any mineral ion concentration in tissues that reduces the dry weight of tissue by about 10% is considered toxic. Such critical concentration vary widely among different macronutrients. The toxicity symptoms are difficult to identify. Toxicity levels for any element also vary for different plant. Many a times excess of an element may inhibit the uptake of another element. For example, the prominent symptom of manganese toxicity is the appearance of brown spots surrounded by chlorotic veins. It is important to know that manganese compete with iron and magnesium for uptake and with magnesium for binding with enzymes. Manganese also inhibit calcium translocation in shoot apex. Therefore, excess of manganese may in fact induce deficiency of iron, magnesium and calcium. Thus, what appear as symptoms of manganese toxicity may actually be the deficiency symptoms of iron, magnesium, and calcium. Can this knowledge be some of importance to a farmer, a gardener, or even for you in your kitchen garden? 12.3 Mechanism of absorption of element. Much of the study of mechanism of absorption of element by plant has been carried out in isolated cell, tissue or organ. These study revealed that the process of absorption can be demarcated into two main phases. In the first phase, an initial rapid uptake of ions into the free space or outer space of cell. The apoplast is a passive in the second phase of uptake. The ions are taken in slowly into the inner space. The symplast of the cell, the passive movement of ions into the apoplast usually occurs through the ion channel. The transmembrane protein that functions as a selective port. On the other hand, the entry or exit of ions to and from the symplast requires the expenditure of metabolic energy, which is an active process. The movement of ions is usually called flux. The inward movement into the cell is influx and the outward movement is called efflux. You have read the aspects of mineral nutrient uptake and translocation in plants in chapter 11. 12.4 Translocation of solute. Mineral salts are translocated through xylem along with the ascending stream of water, which is pulled up through the plant by transpirational pull. Analysis of xylem sap shows the presence of mineral salts in it. Use of radioisotopes of mineral elements also substance the view that they are transported through xylem. You have already discussed the movement of water in xylem in chapter 11. 12.5 Soil as reservoir of essential element. Majority of the nutrients that are essential for the growth and development of plants become available to the roots due to weathering and breakdown of rocks. These processes enrich the soil with dissolved ions and inorganic salt. Since they are derived from the rock minerals, their role in a plant nutrition is referred to as nutrition. Soil consists of a wide variety of substances. Soil not only supplies minerals but also harbors nitrogen fixing bacteria, other microbes, hold water, supply air to the roots, and act as matrix that establish the plant. Since deficiency of essential minerals affect the crop yield, there is often a need for supplying them through fertilizer. Both macronutrient NPK, SETC and micronutrient CUZ and FE, MN, etc. form components of fertilizer and are applied as per need. 12.6 Metabolism of nitrogen. 12.61 Nitrogen cycle. Apart from carbon, hydrogen and oxygen, nitrogen is the most prevalent element in living organisms. Nitrogen is a constituent of amino acids, protein, hormones, chlorophylls and many of the vitamins. Plant Plants compete with microbes for the limited nitrogen that is available in soil. Thus, nitrogen is a limiting nutrient for both natural and agricultural ecosystem. Nitrogen exists as two nitrogen atoms joined by a very strong triple covalent bond, that is N triple bond N. The process of conversion of nitrogen N2 to ammonia is termed as nitrogen fixation. In nature, lighting and ultraviolet radiation provide enough energy to convert nitrogen to nitrogen oxide, NO2, N2O. Industrial combustion, forest fires, automobile exhaust and power generating station are also source of atmospheric nitrogen oxide. Decomposition of organic nitrogen of dead plants and animals into ammonia is called ammonification. Some of these ammonia volatilizes and re-enter the atmosphere, but most of it is converted into nitrate by soil bacteria in the following steps. Ammonia is first oxidized first oxidized to nitrite by the bacteria Nitrosomonas and or Nitrococcus. The nitrite is further oxidized to nitrate with the help of the bacterium Nitrobacter. These steps are called nitrification. These nitrifying bacteria are chemoautotroph. The nitrate thus formed is absorbed by plants and is transported to the leaves. In leaves, it is reduced to form ammonia, then that finally forms the amine group of amino acids. Nitrate present in the soil is also reduced to nitrogen by the process of denitrification. Denitrification is carried by bacteria Pseudomonas and Thiobacillus. 12.62 Biological nitrogen fixation. Very few living organisms can utilize the nitrogen in the form of N2 available abundantly in the air. Only certain prokaryotic species are capable of fixing nitrogen. Reduction of nitrogen to ammonia by living organisms is called biological nitrogen fixation. The enzyme nitrogenase, which is capable of nitrogen reduction, is present exclusively in prokaryotes. Such microbes are called N2 fixers. The nitrogen fixing microbes could be free living or symbiotic. Example of free living nitrogen fixing aerobic microbes are Azotobacter and Bazernikia, while Rhodospirillum in anaerobic and free living. In addition, a number of cyanobacteria such as Anabina and Nostoc are also free living nitrogen fixers. Symbiotic biological nitrogen fixation. Several types of symbiotic biological nitrogen fixing association are known. The most prominent among them is the legume bacteria relationship. A species of rod-shaped rhizobium has such relationship with the roots of several legumes such as alfalfa, sweet clover, sweet pea, lentils, garden pea, broad bean, clover beans, etc. The most common association on roots is as nodules. These nodules are small outgrowth on the root. The microfrankia also produces nitrogen fixing nodules on the roots of non-leguminous plant. Example alnus. Both rhizobium and frankia are free living in soil, but as symbionts can fix atmospheric nitrogen, uproot any one plant of a common pulse just before flowering. You will see near spherical outgrowth on the root. These are nodules. If you cut through them, you will notice that central portion is red or pink. What makes the nodules pink? This is due to presence of leguminous hemoglobin or leg hemoglobin. Nodule formation involves a sequence of multiple interaction between rhizobium and roots of the host plant. Principal stages in the nodule formation are summarized as follows. Rhizobia multiply and colonize the surrounding of roots and get attached to epidermal and root hair cell. The root hairs curl and the bacteria invade the root hair. An infection thread is produced carrying the bacteria into the cortex of the root, where they initiate the nodule formation in the cortex of the root. Then the bacteria are released from the thread into the cell, which leads to the differentiation of specialized nitrogen fixing cell. The nodule thus formed establishes a direct vascular connection with the host for exchange of nutrients. These events are depicted in figure 12.4. The nodule contains all the necessary biochemical components such as the enzyme nitrogenase and leg hemoglobin. The enzyme nitrogenase is a molybdenum iron protein, Fe protein, and catalyzes the conversion of atmospheric nitrogen to ammonia, that is given in figure 12.5. The first stable product of nitrogen fixation is the reaction is as follows. The enzyme nitrogenase is highly sensitive to the molecular oxygen. It requires anaerobic condition. The nodules have adaptation that ensure that the enzyme is protected from oxygen protected protected for protected from oxygen. To protect these enzymes, the nodule formation contains To protect these enzymes, the nodule contain an oxygen scavenger called lag hemoglobin. It is interesting to note that these microbes live as aerobes under free living condition where nitrogenase is not operational, but during nitrogen fixing events they become anaerobic, thus protecting the nitrogenase enzyme. You must have noticed in the above reaction that the ammonia synthesis by nitrogenase requires a very high input of energy, 8 ATP for each NH3 produced. The energy required thus is obtained from the respiration of the host cell. Fate of ammonia. At physiological pH, the ammonia is protonated to form NH4 plus ammonium ion, while most of the plants can assimilate nitrate as well as ammonium ions. The latter is quite toxic to plants and hence cannot accumulate in them. Let us now see how the NH4 plus is used to synthesize amino acids in plant. There are two main ways in which these can take place. First, reductive amination. In this process, ammonia react with alpha ketogluteric acids and form glutamic acid as indicated in the equation given below. Second, transamination. It involves the transfer of amino group from one amino acid to the keto group of a keto acid. Glutamic acid is the main amino acid from which the transfer of NS to the amino group takes place, and other amino acids are formed. Through the transamination, the enzymes transaminase catalyzes all such reaction. For example, the two most important amides, asparagine and glutamine, found in the plants are a structural part of protein. They are formed from two amino acids, namely aspartic acid and glutamic acid, respectively, by addition of another amino group to each. The hydroxyl part of the acid is replaced by another NH2 radical. Since amides contain more nitrogen than the amino acids, they are transported to other parts of the plant via xylem vessels. In addition, along with transpiration stream, the nodules of some plants, example soybean, export the fixed nitrogen as urites. These compounds also have a particularly high nitrogen to carbon ratio. Summary: plants obtain their inorganic nutrient from air, water and soil. Plants absorb a wide variety of mineral elements. Not all the mineral elements that they absorb are required by plant. Out of the more than 105 elements discovered so far, less than 21 are essential and beneficial for normal plant growth and development. The elements required in large quantities are called macronutrients, while those required in less quantity or in trace are termed as micronutrients. These elements are either essential constituents of protein, carbohydrate, fats and nucleic acid etc., and or take part in various metabolic processes. Deficiency of each of these essential elements may lead to symptoms called deficiency symptom: chlorosis, necrosis, stunted growth, impaired cell division etc. are some prominent deficiency symptoms. Plant absorb mineral through roots by either passive or active process. They are carried to all the parts of the organism through xylem along with water transport. Nitrogen is very essential for the sustenance of life. Plants cannot use not atmospheric nitrogen directly, but some of the plant in association with anti-fixing bacteria, especially roots of legumes, can fix this atmospheric nitrogen into biologically usable forms. Nitrogen fixation requires a strong reducing agent and energy in the form of ATP, and to fixation is accomplished with the help of nitrogen fixing microbes, mainly rhizobium, the enzyme nitrogenase, which play an important role in the biological and to fixation is very sensitive to oxygen. Most of the processes take place in anaerobic environment. The energy ATP required is provided by the respiration of the host cell. Ammonia produced following N2 is incorporated into amino acid as the amino group. Thank you friends for listening this audio. If you want any, and please post your comment if you want any kind of improvement. So मिलते हैं next video में. Just stay tuned with Biology Critic and enjoy the knowledge. Okay, bye.